We know that a single cell divides to form two or more cells. The dividing capacity of the cell has led to the evolution of multicellular organisms. Today, there are millions of highly complex species of plants and animals with systematically designed structures. But have you ever thought which structures in the cell might have been involved in the division and multiplication of cells? In this video, we will learn some of these structures which facilitate the cell division process and also perform many other activities. Let's start with centrosomes. Centrosomes are organelles present only in animals. Each centrosome contains two cylindrical structures called centrioles surrounded by amorphous pericentriolar material. The centrioles are aligned perpendicular, that is at the angle of 90 degree to each other. Now, if you look at the screen, you can see that centrioles has cartwheel-like structure. Well, this cartwheel-like organization is made up of 9 evenly placed peripheral fibrils of tubulin proteins. Each peripheral fibril is a triplet and are seen linked with connecting fibrils to the adjacent triplets. The arrangement of these peripheral fibrils create a gap in the center. There is a proteinaceous tubular hub present in the center. The hub is connected to the peripheral triplets by spokes made up of protein. This was all about the structure of centrioles. So, what role do they play in our body? As mentioned earlier, they help in cell division by producing spindle fibers. They also form the basal body of cilia and flagella. The next structure that we are going to discuss is nucleus. Nucleus is not an ordinary structure. It is the control system of a cell. Nucleus was first discovered by Robert Brown in 1831 and later Fleming named the chromatins to the dark strain structure found inside the nucleus. What else is inside the nucleus? Let's see. Nucleus is bound by a double membrane, that is nuclear membrane, with a space in between called perinuclear space. The nuclear membrane acts as a barrier separating the nuclear environment and the cytoplasm. Inside the nucleus, the fluid matrix is known as nucleoplasm. Nucleoplasm has chromatin fibers, nucleolus and nuclear proteins in it. We'll now see the detailed structure of a nuclear membrane. As said earlier, nuclear membrane is double layered. The outer nuclear membrane is seen in continuous with the endoplasmic reticulum. Ribosomes are also seen bound with the outer nuclear membrane. The nuclear membrane is not continuous in many places. It has minute pores called nuclear pores. These pores act as a passage for RNA and protein to move in and out of the nucleus. In case of nucleoplasm, it is a fluid matrix containing nucleolus and chromatin fibers. Nucleolus is a non-membrane spherical shaped structure. It is the site for ribosomal RNA synthesis. In cells which are highly active in the synthesis of protein have larger and more nucleoli. Coming to the chromatin part, a cell in its interface will have uncoiled thread-like structures called chromatins. But when the cell enters the phase of cell division, the chromatin fiber get condensed to form chromosome. If we visualize the chromatin fibers under electron microscope, we'll be able to find a chromatin fiber contains DNA, some basic proteins called histone and non-histone proteins, and also RNA. When a cell is ready for division, chromosome is formed by condensation of chromatin fibers. Each chromosome has two chromatids separated by primary constriction called centromere. The centromere has dish-shaped structure called kinetochore. It is to the kinetochore that the spindle fibers attach in metaphase. Do you know the different types of chromosome based on the position of centromere? A chromosome is said to be metacentric if the centromere is at the center and has two equal length chromatids on either side. It is said submetacentric if the centromere is not at the center and one chromatid is slightly longer than the other. When one chromatid is very long and the other very short, it is acrocentric. Here the centromere is situated near the end. The last type is telocentric, where there is one chromatid as the centromere is at the tip. In few chromosomes, there is a small non-staining constriction at the end called secondary constriction. The terminal part above the secondary constriction is the satellite, and chromosome with satellites are called sat chromosome or satellite chromosome. Let's now see the last structure of this session, that is microbodies. Microbodies are membrane-borne minute structure that contain various enzymes. These microbodies are present both in plants and animals. The different microbodies include peroxisomes, glyoxysomes, etc. We have now come to the end of this session. Can you find out what are the roles played by the microbodies such as peroxisomes and glyoxysomes? Let's now summarize what we have learned.
Manorama Horizon. Learning made simple.